Today we will learn about how we can make bacterial smear for doing any kind of staining. For bacterial smear preparation, we require 1 drop of water. We require a dropper plus Bunsen burner bacterial culture and inoculation loop for introducing bacterial culture into glass slide. First, we will put 1 drop of water into glass slide like this with the help of dropper. We will put 1 drop of water into glass slide like this, and after that we will red hot the inoculation loop into Bunsen burner like this. You can see, guys. And after that we will cool the inoculation loop like this. With the help of inoculation loop, we will collect with the help of inoculation loop we will collect bacterial colonies from the bacterial culture. With the help of inoculation loop, we will collect the bacterial colonies and we will put these colonies into glass slide and we will make bacterial smear like this. We will mix these bacterial colonies into glass slide like this. We will mix these colonies into glass slide like this and make a thin smear like this. You can see, guys, we will make a smear, thin smear like this. You can see. Now we will heat fix the smear in Bunsen burner in forward and backward motion like this. You can see, guys. And in this way we will make a bacterial smear. You can see, guys. And after preparing bacterial smear, we will do any kind of staining and visualize these bacterial cells under the microscope, guys.